the screen.
Yes, I'm seeing the screen now.
So I'm Dr. Lee, Melissa Lee. This is my background. I have a PhD in microbiology, and I'm also a medical doctor. You want to come back to Jamaica?
Yes, definitely. Yeah, I really want to contribute.
You have to be prepared for a lot of Jamaican way of doing things, a lot of resistance. It's also very stressful being at home and like having achieved it. If you actually, for example, if you do it now at 21, by the time you're 26, 27, you should be done your PhD, right? Because the master's MPhil should take you 2 years. PhD shouldn't take you more than 3 years. In this day and age, it shouldn't. If you're done by 26, 27, and you decide to come back home, just prepare for a lot of resistance.
Yeah.
Think that you're needed because you're doing something that is unique or not very common in such a small country. But you would think that added value would be a bit more welcoming. So you will have to decide or be dedicated to what you do and know what your purpose is.
Like you have to build from the ground up.
Exactly. But I would say continue doing it. It's exceptionalism. People sometimes don't like it, but it is what it is.
Because I know like abroad, they do have like advanced machinery and the methods and the type of research that they're doing as well.
It's not necessarily even more advanced machinery. We actually have everything here. Surprisingly, you would think that we don't have. We probably have almost everything that is advanced or can access it easily. It's just that if the mindset is not there yet, it feels like it's not there. But for example, if you want to do something in cellular engineering and you want to incorporate some ideas, just researching around it, you'll be surprised. The idea is to actually try to build the lab here, get everything that we want so that we can have students access anything that they want, because I primarily was just disadvantaged as a young person, not being able to access what I wanted in my own country, even though it was here. The idea is to emerge that field that is already emerging. So the molecular engineering, where we can make our mRNA vaccine, where we can do chemical engineering, which is molecular engineering, for treatment, for anything at all. I think we need to start looking in that direction. So I guess I can start the session now. I have a no-fail policy, meaning that I don't believe in failure, not because people are dumb, but because I don't believe that I need to make it hard for people to fail. So definitely it's not going to be a course that you need to worry about failing unless you're not doing your work, really not doing anything at all. Like for you guys to at least come into the lab every week or other week to do like random quizzes and stuff like that that will contribute.
Final grade.
Yeah. If your background is not noisy, you can leave your mic open and you can interrupt me at any time. It's not that serious. Morning, Ryan.
I'm sorry I joined a bit late. I'm actually on the road right now driving.
Okay, that's fine.
Yeah, so I just wanted to ask, since I joined a bit late, was there anything that was discussed before I joined that I should know?
No, I was just basically giving advice on future studies and nothing that I would consider important if you're not interested in pursuing your master's PhD afterwards.
Okay, so you're saying that the Wednesdays then would be our theory sessions, right? Is lab?
Yeah. So like the Monday, definitely the 9 to whatever. No, I know you guys don't want to see my face on a Monday morning, right? Friday session, your flexible session, where that's your period where you just come in. I'll actually have stuff prepared in terms of the practical sense of it. Stuff prepared so you could come in, ask a question. It might not be the most ideal now because, as I said, the lab is being constructed as we speak, and nothing too stressful, not too complicated. I don't like stress. I'm not going to stress you out. If I'm stressing you out, please let me know.
All right, will do.
Yes. So if you see 30 slides, count that as 1 hour. If you see 60 slides, count that as 2 hours. So for today, I have like 60 something slides, and it's basically a synopsis of everything everything that you will cover for the semester. In terms of the course assessment, it will be continuous. So for example, if you come in next week, whatever I give you, it's like an assessment. I'll try to prepare you mentally for everything so you know what I'm expecting. So when you come here and you do the small quiz, that contributes to your final grade, or to the 40% that I see that, you know, contributes to some of the final grade.
Will we be getting like mid-semesters then, or no?
No, so you're not getting mid-semester. You'll just be continuously assessed. For example, next week, I tell you from now, you will need to know like gram stain, the procedure. You will just need to be able to know how to do it in practice. Identify, for example, a gram negative from a gram positive. If you guys have no other concern, then I'll just start with the content itself. The course is cell and tissue mechanics. It's an introduction. Everything has a starting point. We like to assume that as human beings, right? Because it's only logical for us to process, because we just don't know why we're here. We don't know when we arrive. We're just trying to figure out stuff. So we try to put a start point to everything, which is why my PowerPoint starts with pre-Cambrian era, which basically looks at a geologic period in time that was before any cell was ever formed, or that's what we think. Over 6 million years ago, they said that the first cell was formed. This first cell should be what we call a prokaryotic cell. So you have different types of cells. So organism, on the other hand, can be single cellular or can be multicellular. For example, you're a giant multicellular organism. Parasites can be single cellular, multicellular. So as long as you get the concept that, you know, cell can be single, it can be multiple. With cells coming together, they create networks and form tissues. The structure, the function of the cell creates a biological unit. So what's important for this course is to understand the structure and the function. You know that a group of cells come together, they form tissue. You should at least try to understand the function of each. So as I said before, cells can be broadly categorized as prokaryotic, eukaryotic. On the prokaryotic, bacteria. So for example, the prokaryotic cells, the typical features, what exactly are you expecting? And the more you do this, the more you see, the more you realize that almost everything is the same. So even though they tell you a bacterial cell, if you look at the human cell or a plant cell, it's pretty much almost similar structure, composition. For example, it either has a cell wall, probably an outer protective coat, something that protects it from the harshness of the environment. So for example, if you look at the prokaryotic cell, for example, this bacteria here, you see pili. Some of them you'll see that they have pili, these finger-like projection that's just sticking off of it. And these pili, they have different function in different cells in different environments. Flagellum, for example, these are external appendages. They have specific function. For example, anybody knows the function of flagellum?
Movement of the cell.
Exactly, movement of the cell, easy stuff, right? And are you able to tell me of any cell with a flagellum? Not bacteria, but be more specific.
And I learned this in grade 2, but I completely forgot.
So think about you being a male.
Oh, like a sperm, yeah.
Exactly. So you have sperm cell. Your sperm cell has a flagellum for motility. Then we also have the cytoplasm, ribosomes, nucleoid, or nucleocapsid. These tend to contain the genetic information, the DNA, RNA, that has the genetic instruction for making protein or sustaining the survival of the cell itself. Anybody has an idea what the ribosomes, the functions of the ribosomes are? Sometimes when you get a wrong answer, it feels horrible. But the feeling is what creates the memory, and the memory is what will make you learn. Animal and plant cells generally are eukaryotic. And if you look at the organelles, everything that comprises these cells, so what you will need to do for this course, know the distinct differences between them. So at least know the difference between a prokaryotic versus a eukaryotic. Know the difference between a plant cell and an animal cell. Know which one has a cell cell membrane or which one doesn't have a cell wall, or which basically just stuff like that. And the differences are very small. For example, the plant cell has a chloroplast. Anybody know what the function of the chloroplast is?
Chloroplast is used to convert light energy into carbohydrates.
Very good. For photosynthesis, for energy production. Anybody know the function of the mitochondria?
The powerhouse of the cell.
Powerhouse of the cell. Can you believe that when I went to the US Embassy a few years ago, that was a question?
Really?
Yes. I was at the US Embassy. That was the most random thing ever. I was just at the interview and I was supposed to be a microbiologist, and the person was just like, what is considered the powerhouse of the cell? Yeah, a pretty random question. So definitely this is for ATP generation or to provide energy. What's the difference between the nucleus in the plant cell versus the animal cell? Any big difference? Not really. Not really. You have the envelope, a nuclear envelope, and pore. And why would you have a pore?
I know one function would be to allow the passage of genetic information out of the nucleus. And then the next one, I know that you have some hormones that actually initiate enzyme cascades. So you can also have enzymes that enter the nucleus via the pores as well, like initiate certain metabolic processes. And
Exactly. If I ask you a question, you already know the answer. And this is the approach that I want to take. Yeah, you know the answer long before I do. You know why? You have Google, you have your cell phone. And I think I did well with many things because I understood this very early. You're a technology baby. You know everything because everything that you need to know is in your hand. And that's the thing. There are a lot of things outside our control as human beings, and I try to be realistic about it. For example, if you're in the workplace, what matters at the end of the day is your productivity, your efficiency, or your way of doing things effectively. So we have ribosomes for everything. For example, the prokaryotic, you have the ribosomes. And if you look closely everywhere, you'll find ribosomes in basically almost everything. Make the comparisons and you will learn by just looking at pictures. You don't need to do much. So we have the smooth endoplasmic reticulum, rough endoplasmic reticulum. You see the smooth endoplasmic reticulum here in the plant cell, and we also have it in the animal cell as well. There are also protein, there are structural proteins, functional protein that help with movement, and many other things. For example, you're looking for the microfilaments, microtubules. As I said, the prokaryotic cells are presumed to be the first cells that were ever formed. Looking at the prokaryotic cell here, difference between the bacterial cell wall and the bacterial cell and the eukaryotic cell. Do you see a cell wall here on the eukaryotic cell?
No, not on this one. This one will be an animal cell.
So you have a cell membrane though, right? There's a cell membrane. And again, mitochondria, the chloroplast. So this looks like what? A plant cell, right? With the chloroplast.
Oh, yes.
And if I ask you for an example of a bacteria with flagella, are you able to tell me that? Or a flagellum? No Google? Internet too slow?
Yeah, man, I move slow, yes.
Okay. So this is a broader way of classifying cells. Eukaryotic cells, we know that comprises your plant and your animal cells already, your fungi, and the protista, kingdom of different microorganisms. Bacterial cells already, we know, are prokaryotic. So that's the take home. Typical prokaryotic cell contains cell membrane. Bacteria, they have their cell wall, cell membrane, chromosomal DNA that is concentrated in nucleoid. They all have ribosomes as well as the cell wall. So they have a cell wall, a very complex cell wall. And the importance of the lab session would be for you to understand the importance of the cell wall itself. Because when you do a gram stain, you're staining the cell wall of the bacterial cell. And when you do, it helps you to differentiate the type of bacteria that you're working with. Bacteria can be gram negative or they can be gram positive based on the cell wall composition. So for example, if you look at E. coli, a bacteria, Google should tell you that E. coli coli has like pili, flagella that helps it to move. E. coli is a bacteria that typically cause urinary tract infection. So if you're a female, you've gotten urinary tract infection in the past, likely to be E. coli. If you're a male and you had urinary tract infections, also likely to be E. coli. It's mostly due to E. coli. Why? That bacteria is from a big family of bacteria. They call them Enterobacteriae. They're gram-negative. So when you come in and you actually stain the cell, you will know the difference between a gram-negative and a gram-positive. When you come to the lab session, I can give you a plate and you can have a bacteria on it, and you can just take that bacteria off, put it on the glass slide, stain it, and you can tell me what bacteria that is. And that bacteria could possibly be coming from a patient's urine. You're not a physician, but at least you'll become one by the end of this, hopefully. For bacteria with pili, fimbriae, flagellum, E. coli is a common example. If I, for example, ask you a question or example of bacteria with pili, fimbriae, flagella. Plasmid is a different thing, if I should mention that. Common prokaryotic cell shape. As I was saying about the cells and the shape, how do you differentiate the bacterial cells? Not all of them are the same. Just like me and you, we're not the same. We don't look alike. So there has to be some way of determining why I am different from you. If you go to the molecular level, which is why I try to advance this discipline. If you go to the molecular level, there is almost nothing to see, but there is so much that you can learn. There is so much that you can know from just studying things at the molecular level. Common prokaryotic cell shape, as I said, you have the coccus. Let's talk about the bacteria, because we know bacteria can be different shape based on how they look, morphology. So it can be coccus, which means that it's round. Staphylococcus aureus is a bacteria that bacteria you find in so many patients coming with blood infection, that bacteria is actually a normal bacteria on your skin, Staphylococcus. So these coccus or cocci, round shaping bacteria, they're all over your nose right now. There's a statement that says that there are more bacteria in your mouth than there are people on this earth. Who can tell me how many people on this earth estimated?
Isn't it almost like nearly 8 billion?
Imagine you having 8 billion bacteria in your mouth. You believe that?
I mean, they're very small, so a lot of them can fit in a small area.
People would just look at that and think, wow, ew, nasty. No, we all have to live symbiotically with these microorganisms. So the cocci, gram-positive, they're round. So when you look through the microscope, it won't look like this. This would look more like an electron micrograph where you magnify it way, like much bigger. What you will use to determine what it is the color that it retains when you do the stain. Come into the lab and you stain it, it will retain either red or purple depending on the type of bacteria that it is. And that's what you will learn and that's what you'll understand. So other bacteria, prokaryotic cell shape bacteria types can be bacillus or rod-like. There is a bacteria called Bacillus cereus. You ever eat rice yet and have food poisoning? Anybody can recall?
Food poisoning from rice?
Yeah.
I haven't said that.
If, for example, you know of somebody who's having diarrhea, for example, somebody just come and say, yo, I have diarrhea after eating this thing, you can ask them to eat rice. If they eat rice, you know it's probably likely to be a bacteria called Bacillus cereus. It's a rod-shaped bacteria. Or it can be the same Staphylococcus aureus. It's the same bacteria that you find on your skin. You have different types of these bacteria that you can even just find on chicken. For example, if somebody eats fried chicken, you can be the doctor to intervene and say, wow, this sounds like Staphylococcus aureus. Or the engineer that says that, what can we engineer to prevent Staph aureus from growing on your chicken?
Mm-hmm.
And then we have the curve or rod-shaped ones, Vibrio cholera. You know of cholera, right?
Yes.
It's like a rod. It's like a little comma. You have diarrhea as well. Them say rice water stool. They can also be coccobacillus, as in they can have a combination of round shape and rod-like. They can also be spiral. Anybody know of a spirochete? Google in action. Slide.
Ebola.
Come again. That people get when they have unprotected sex. Start with S too.
N?
S. It start with S.
Syphilis.
Perfect. So you see this big picture that I have here on the right is basically what it looks like when they magnify it on the electron microscope. micrograph. Syphilis. This part of it, I'll focus a bit more with subsequent PowerPoints in terms of the DNA inside of the cell, the organization of the DNA and packaging in the chromosome, and even a little bit more about cell replication. What's important to note is that bacteria, you basically have functional protein and it differs based on the type of cell. This is basically a micrograph of a nucleoid, of the nucleoid region of a bacteria, and it basically is showing the dense area. This is just shifting the focus or zooming in now on the eukaryotic cells. So the two types of eukaryotic cells, the animal cells, the plant cell. And as you can see, both of them have a cell membrane, both of them have lysosome. Anybody know what the function of the lysosome is in the cell?
Generally, carries hydrolytic enzymes, I think. Or just for transport, I think. Transport of things. So you can have like endocytosis and exocytosis. And yeah, sometimes you use them for like hydrolytic decomposition of certain compounds that may be hazardous.
Well, you sound like the scientists. Like you like the detail, that's good. Or you're very good at Googling.
Oh no, that's not the case.
Organelles. Organelle is a biological structure that performs distinctive function inside the cell. So as you know, all of these things inside of the cells, they have their own function. So they're not just there. It's just like your organs in your body. They all have specific function. Well, 99% of them, because some are rudimentary. For example, your appendix. Well, claim that we don't know what the appendix is for. No, like in this modern intelligent man. So they're thinking it's like it has like somewhat of a primitive function where it has no real function in us. Some people are knowledgeable about appendix and the probability of appendicitis and the complications of it. Them actually just take out the appendix of the baby when the baby born. You know, like how you circumcise them, pretend to be the intelligent man and take out the appendix. And even some of your tonsils, the adenoids, they're just there. Some say they don't have any function, but they have lymphatic tissues, as in they have white blood cells and cells that help to fight infection. And so is the appendix. They have a lot of lymphoid tissues in the appendix. So sure, I mean, if you take it out, you don't really need it. You can live without it, but I'm assuming that lymphoid tissues are there for a specific reason. Organelles literally mean little organs. So these things in the cell are little organs, just like we have big organs in our body. They all have function. And the eukaryotic cell, just know the diagram by detail. I find a picture where I can just slap on till I just take off all of these labeling and just have you fill them in. That would be a nice quiz. For some of them, yes, you might have to fill it in, but some of them I might not ask you what it is. I might just ask you what the function is. So it's not only to know what is in the cell, but also know the function. If you see a slide that is highlighted with anything that is in red, it just means that you need to know it. Because if I go and ask anything in exam or final exam, it's just going to be something that is highlighted in red or something that I say to you in the session. So we've been talking about the cell membrane being bounded, housing the organelle as well as in the cytoplasm. So it's important to know what the function of the nucleus, the endoplasmic reticulum, the Golgi apparatus, the mitochondria. If you want to further your career in anything at all, especially if it's biomedical engineering and engineering, you will work a lot with cells. So it's important to really know the anatomy of the cell. So this is my Google Scholar session where I'm going to just ask you the function of these. You can Google them. Whoever give me the best Google answer, that's how we score the point. So anybody can tell me the function of the perioxosome. So you're here to teach me as well.
They're involved in oxidative reactions.
Metabolism.
Yeah, metabolism, yeah.
You break down fatty acids.
Internet moving slow.
Wow, you failed the Google Scholar exam, man. How about synthesis, assisting the synthesis of cholesterol? So the perioxosome helps cholesterol synthesis, bile acid synthesis, for example, in the liver, just a random example in human, breaks down excess DNA product, purine, pyrimidine. But if I ask you anything about perioxosome, for example, where the animal cell is concerned, it would just be to relate it to the human liver.
Specific to the liver.
Yeah, so the mitochondria function. Any specific function?
It's a site of oxidative phosphorylation.
So what that means?
So that's basically the process by which you have the formation of ATP, the transport of hydrogen ions across the inner mitochondrial membrane. And then that will then result in, you'll actually have the oxygen now, which you actually take in by breathing. That's combined with those hydrogen ions to form water. But yeah, you just want the formation of ATP to take place so that it can be used by the cell.
How about the Golgi complex?
It's used for protein processing and lipid processing.
How about the microfilament? What's the function of a microfilament in a...
It's involved in the cytoskeleton.
So it's a part of the cytoskeletal structure.
I think essential, especially when it comes to like mitosis, where you have the filaments that actually pull the mitochondria apart into the separate cells.
How about its function in a muscle cell or skeletal muscle? Because it helps with cell movement.
Yes.
So it helps with muscle contraction as well. Microtubules. Everything that I'm emphasizing, it's stuff that you will hear over and over. Things will get easier. How about the microtubule?
I know they're involved in, again, they are part of the cytoskeleton, so they help to give the cell its shape.
So those are the important things that we want to take from this one. Anatomy of the cell, basically showing the same thing. So in terms of the cell wall or the membrane composition, I would suggest know the details where this is concerned. The structure, the lipid composition, for example, you will know that cell membrane of phospholipid bilayer, some areas are hydrophilic, certain areas are hydrophobic, and you have proteins, different types of proteins with different functions. So as we were talking about the bacterial cell wall, as I said before, the bacterial cell can be gram-negative, can be gram-positive, and it has what we call peptidoglycan layer. So when you do the gram stain, that's where you'll stain. You'll stain the peptidoglycan if, for example, it's a gram-positive bacteria. And that is a composition of the bacterial cell wall that you're staining. So if you look at the gram-negative, and that's a little bit different in terms of the cell wall, way different from the gram-positive. So if you should know anything about gram-negative and gram-positive cells or bacteria, the difference in the structure of the cell wall, that is a big difference. They definitely will need to know, for example, lipopolysaccharide, O polysaccharide, 4 polysaccharide, lipid A. Those things that will stand out and that will make the difference between gram-negative versus a gram-positive. For example, if you look at this gram-negative bacterial cell, you have lipopolysaccharide. Same E. coli that I told you about, it's a gram-negative bacteria. Same one that causes a lot of urinary tract infection. It's a gram-negative bacteria. It doesn't only cause urinary tract infection, it can cause food poisoning as well. You have E. coli, you have different types of E. coli strain like 015, enterohemorrhagic, toxigenic E. coli, different types that can cause really bad diarrhea, can cause bloody diarrhea, can cause blood in the stool, can cause even diseases that are not necessarily, doesn't have anything to do with the gastrointestinal system. For example, the same E. coli can cause what we call hemolytic uremic syndrome in a child, where it's just damaging their kidney, it's just blood in the stool, and they just have blood in the urine because it's damaging the kidney and it's also damaging the gastrointestinal cells. So the important takeaway from the gram-negative bacteria is the lipopolysaccharide, the O polysaccharide, the 4 polysaccharide, the lipid A, they are a part of the cell wall and they are important virulence factor. What we call virulence is like an antigenic property. So you know when they talk about immunology, they say antigen or antibodies. The antigen is what you find on the cell. For example, lipopolysaccharide would be the antigen on the E. coli that is causing you to have a problem. So your antibody will respond to this lipopolysaccharide because it sees that part of the cell wall as the foreigner. So it's usually something that the body picks up on the outer surface of the cell that creates a trigger or an alarm in your body that says that this is a foreigner or this is bad. So that's it for the gram-negative. This is just a picture showing the difference between the gram-negative and the gram-positive. You have the outer membrane, you have the lipoproteins, both of them have the peptidoglycan. When you stain the gram-positive, the thing will be trapped in the peptidoglycan layer, the stain, and it will just retain the dye itself. So for example, if the dye is purple, the cell wall will stain purple. So when you look through the microscope, you're going to see purple cells. And if you see purple cells, what will be a gram-positive? When you stain the gram-negative, certain part of it that also retain the dye, and this retains the red dye. So for example, if you stain a cell, you're going to put two dyes on it. You're going to put, for example, a purple dye and a red dye. And if you see purple, you know it's gram-positive. If you see red, you know it's gram-negative. And if I ask you for an example of a gram-negative bacteria with pili or fimbriae or whatever it is, you can already tell me E. coli. And that's a common bacteria that causes a lot of things. This is the gram stain procedure. As I was just explaining a while ago, the primary stain is crystal violet. So you're going to stain it this purple color. You're going to put some iodine on it, and then you're going to add something to decolorize it. For example, if you put the crystal violet on it and the peptidoglycan layer doesn't trap this crystal violet, when you add the acetone or the alcohol to it, it will just wash off because it's not trapped in the cell. So when you put the counter stain, which is safranin, that counter stain is red. So if the counter stain is on it and it stains red, would you be able to tell me which is it? Would it be gram-negative or gram-positive?
Negative.
Gram-negative, exactly. So this is just basically the staining procedure, exactly what you do when you do a gram stain. Gram-positive cell walls have single membrane enclosed by thick cross peptidoglycan. Thick peptidoglycan will take up the dye. So when the peptidoglycan layer take up the dye, it appears as purple. And I'll just go back to show you the peptidoglycan layer. You can see it here in the gram-negative. You can also see it in the gram-positive. But you realize the difference between the peptidoglycan layer in the gram-negative and the gram-positive?
Yes, miss.
Yes, there's a big difference here. Thick peptidoglycan layer will take up the dye, and the dye will adhere to the layer. And then, as I said, you can use the alcohol or acetone, and that will wash out the dye. But if it is not a gram-positive, so the dye will just be absorbed into the peptidoglycan layer. You will have to heat the slide. So in terms of the procedure, you heat the slide, you will put the crystal violet on it. It's nothing special. So what you will have is like a glass slide, a little square glass slide, and you'll have like blood agar plate with the bacteria on it that you can already see. You obviously won't know what bacteria it is until you take the bacteria off the plate and then you put it on the slide. So you heat the slide first, then you stain it with a crystal violet, and you leave that for 60 seconds. And then after that, you add the iodine, you leave that for 60 seconds, then you rinse off the slide itself. You're going to rinse it with acetone or alcohol for 20 seconds. You're just going to put it over it, and that's after it dry, right? Then you're going to put the counter stain on it that is safranin, and that will basically tell you whether it's negative or positive. So gram-negative cell wall have a thick layer of peptidoglycan in the periplasmic space within it. It's inner and outer lipid membrane. So as you can see, the peptidoglycan layer is thinner in the gram-negative and it's much thicker in the gram-positive. So that's basically all you need to know for the next quiz session. So in terms of the cell wall, we're looking at different cell walls here, right? So you're looking at the cell wall of the fungi. The only difference between the fungi or fungi, I think is plural. The fungi has chitin. So if I ask you what's the difference between the fungal cell wall versus a bacterial cell wall, you can see the difference here. You have the peptidoglycan layer that is arranged differently in the gram-positive. It's a bit thicker in the gram-positive versus the gram-negative cell. You have the LPS, the lipopolysaccharide for the gram-negative. So just know these words, the peptidoic acid and lipoteichoic acids, those make up the peptidoglycan layer of the gram-positive. The gram-negative has a peptidoglycan layer, but you already know it's thinner. And for the fungi, they don't have peptidoglycan layer. What they have is chitin. Oh, you also have the monoproteins for the fungi. So those are the important things to remember. You have a lot of different ways to see cells, identify the morphology of the cells. In reality, in practice, it's best to actually learn the morphology, learn the stains, and once you know what the stain is doing, what it's trapping in the wall or the membrane or whatever it is, it's easier to understand the structure and the morphology of the cells. And there are many other ways you can stain cells. So gram stain is just one. You have lactophenol, that's one. You have India ink. You have a million different ways to stain different type of cell based on the structural composition of the cell. The virus, no, it's not a cell, but we have to make that clear, which is why it's just randomly appearing here. They're acellular. It's not a cell. It have no nucleus, cytoplasm, or organelle. For coronavirus that's going around, it's not a cell, it's just protein particles that is just waiting for your cell to use the organelles in your cell, inside your cell for its benefit. So if the virus wants to replicate, it no need to have no organelles. It's a obligate intracellular parasite. You know, like some people, them kind of just depend on you for everything, to live, to breathe. That's literally what the viruses are. Depend on you, or multicellular organism with organelles to sustain their existence. If corona should survive, it's only surviving because it's jumping from one cell to the next. So if it dies in one person, it has to jump to another person and make multiple copies of itself in order to sustain its existence. And you can't blame coronavirus. All cells are designed for survival. Everything in your body is just primed for survival. It's just automatic. It's just genetic coding by default. Viruses are, to me, intelligent because it doesn't need to have all of that baggage to be able to replicate. Viruses consist of two main parts, nucleic acid. Nucleic acid can be DNA or RNA. It can be a DNA virus or it can be an RNA virus. Like, for example, coronavirus is an RNA virus. You also have capsid. So it's like when you have the cell membrane, viruses can have like capsid or envelope protein. Protein that basically surround the material. This is just basically showing that it doesn't necessarily have to have the same thing or be made up of the same composition, as long as, for example, it has a function that works. Capsid is protecting the environment of the virus. Viruses can have an envelope or capsid. And this is just showing the structure of the virus. This is the HIV virus. And what's this? Capsid, the bacteriophage. Okay. If you should know anything about viruses, you should know that it's not a cell. There's a reason why you learn about cells and tissues, because you need to understand the details of cells. This capsid that surround the virus protein coat is protecting the genetic material, DNA or RNA. So the viruses, in my opinion, I think viruses are far more intelligent than us, because if they see that all you need is the genetic information to carry out the function, you don't need the baggage. Thing is, if you never have kidney, you couldn't have kidney disease. If you never have a heart, you couldn't have heart disease. Viruses do not have ribosomes, mitochondria, or other organelles. Makes sense, right? Why wouldn't have ribosome? It makes sense, right?
Yes.
Because it just uses whatever you have to carry out its functions. At your expense, I will make many of my cells. Isn't that selfish? Well, in human terms, it is. For biological terms, savage and genetically superior. Another important thing that we'll look at, especially doing this course, membrane transport mechanism. It's a cell. Yes, we know there's a membrane or there's a cell wall. Things have to go through, in and out, or be within or around the environment. It's only logical that you need to understand the mechanism of transport of things in a cell. Simple diffusion. Anybody know the definition to that? Google Scholar.
Diffusion. It's just movement of particles from an area of high concentration to low concentration.
Exactly. What's the difference between simple and facilitated diffusion?
Well, it's still the movement from high concentration to low concentration, but in order to achieve the transport across the membrane, you need the help of transport proteins.
Perfect. So you have the channel proteins here. About active transport versus passive transport. Anybody know that?
Active transport is the transport of molecules from an area from low concentration to high concentration. But because there's like you have to go up the concentration gradient, there's an energy expenditure. So typically it requires ATP.
Exactly. So for example, sodium and potassium to move in and out of the cell, it needs what? ATP, right? So you would do well in medicine if you just understand the basic things, because a lot of things in medicine, for example, sodium, potassium. If your sodium is high, it's called hypernatremia. Potassium is high, hyperkalemia, hypokalemia if it's low, hyponatremia if it's low. Sometimes, for example, patients with diabetes, if there's imbalance in their potassium level where it gets really high, you will have to give them something to create that shift where you want for example, the sodium or potassium to run into the cell, because sometimes the extracellular concentration is so high and you want it to go back into the cell. All of that requires energy. So with patients with even diabetes, you can understand how you correlate ATP, energy expenditure, the use of sugar, and the movement of electrolytes. We call them electrolytes: sodium, potassium, chloride, all of these ions. Basically, energy, ATP, is necessary for them to actually move in and out of the cell. In terms of the anatomy now of the human cell, we'll also need to know that. So we'll look at the bacterial cell, bacteria, virus, fungus. We know that virus is not a cell. We know the broad category already, the eukaryotic versus prokaryotic. We just need to know what inside of the cells and what are the function of these things, and the difference between a bacterial cell, a fungal cell, of all cells. You just need to know them. Also, the animal cell itself. You don't need to know the difference between the animal and the plant. So we'll also have to look in detail at the animal cell, which is the human cell. You will eventually understand that you have different types of cells in the body. It can be stem cells, fat cells, sex cells, bone cells, skin cells, blood cells, nerve cells, pancreatic cells, cancer cells. You know that cancer cells are your cells, or for example, they can be blood cells, bone cells, skin cells that just go through genetic mutation and proliferate or multiply uncontrollably. It's just undergone genetic mutation. Also have endothelial cells. These cells tend to line your blood vessels. They're all lined by endothelial cells, and also the muscle cells. So you should be able to have a good in-depth knowledge as to all the cells in the body, their function, how they differ. In embryology, you should know the difference between ectoderm, mesoderm, and endoderm. During your development, for fertilization, they come together and then they start dividing. Few hours, it's 4 cells, 8 cells. They go through different stages. What is important is during this embryologic period, you need to understand that there are different cell types that are formed. Different cell types have different function, and that will determine what they develop into. For example, the ectoderm during the embryologic period will eventually produce your skin and nerve cells. So ecto meaning outside. If I say somebody's an ectoparasite, it's a parasite that lives outside of you, and you actually do have ectoparasite. I'm sure you have friends who are classic ectoparasite. Anything that's ecto or outside, like skin, that comes from ectoderm. Nerve tissue, a bit difficult to remember that it's ecto. It's basically on the outside, but within the central nervous system. Mesoderm give rise to anything in the middle, like just remember middle. For example, the bones and the muscles, they're middle, like they're between the skin. So the mesoderm comes from bones and tissue. So if I ask you a question, I might ask you example of something that derived from mesodermal origin. Endoderm give rise to the liver, tend to give rise to the internal organs or the stuff that cover the organs, so like the liver, thyroid are example of endoderm. In mammals, approximately 50 to 150 cells make up the inner cell mass, and that was basically what I was trying to explain a while ago. During the embryologic period, when your mother ovum or egg is fertilized by the sperm, you have what is called an inner cell mass. This is basically around, when it gets to about 50 to 100 cells, it's like it creates an inner cell mass, and this is basically during a stage we call blastocyst stage, and this is around day 5 to 14. That imagine that's literally 5 days after fertilization, there are so many cells that are there. During the development, they subsequently differentiate into all body types, so they give rise to pluripotent stem cell, and that's how you have endoderm, mesoderm, ectoderm. So they get some genetic signal, and I always say the molecular aspect is important to look at, because there are specific genes that give the instruction automatically for the cells to be differentiated into what they need to be differentiated into. So they are just responding to signal in the environment. What creates this signal? What is the godlike structure in man, or this message or signal myelin in man that give these genes the instruction to create these cells. We're yet to figure that out. I guess that's why some of us don't think that we're God, even though some of us would like to think that we're God-like. The process is initiated by the differentiation into 3 germ layers, and these are the ectoderm, as we already noted. It gives rise to the pigmented cell, or the out cells on the outer layer, like your epidermis, like your entire skin is from ectodermal origin, your nerve cells as well. And the mesoderms give rise to the muscle, things within, like, or in the middle, the blood, kidney cells. And endoderm is responsible for certain immune cells, lung cells, pancreatic cells, thymus, and other organs. In terms of the stem cell or pluripotent stem cell, they differentiate, they undergo differentiation, a process in the bone marrow. For example, the cells in your body, the immature ones come from progenitor cells in the bone marrow. So these stem cells can differentiate into erythrocytes, which are the red blood cell, white blood cell. So they can give rise to red blood cell or white blood cell. Red blood cell here is your erythrocyte. White blood cells are your monocytes. These are white blood cells that circulate in your blood. The neutrophils, these are also white blood cells that you find in a lot of your organs and tissues. The basophils are another type of white blood cell that you can find in your tissues as well, or the blood. These basophils help to fight specific infection. For example, each white blood cell have a different function. They were intelligently designed. The monocytes, they're just parading in your blood to eat up anything, any bacteria. So for example, like the E. coli comes into your bloodstream, it recognizes polysaccharides from the lipopolysaccharide from, for example, a gram-negative bacteria. The monocytes will recognize it and try to eat it up. The eosinophils, they recognize anything that is parasitic. The basophils, for example, if you have allergic type problems, it's probably due to these cells just producing a lot of things from within it that will trigger like a reaction for you. Of course, you also have different progenitor cells that develop into. So it can be white blood cells, can be red blood cells, and it can be lymphoid progenitor cells. These are just different types of white blood cells. The B cells that are made in bone marrow, and the T cells that are made in the thymus. So this is just basically another diagram showing exactly what I said just now. The only thing is, for example, these cells, before they are fully synthesized or created, in the young stage, they are known as blast cells. So anything that is blast, it's a younger version of the cell. They call them myeloblast cells. For example, the neutrophils, the young neutrophil, blast cell. Anything that is a young version of the older version of the cell, you call it blast. For example, if you go to the bone marrow, the cells in the bone marrow that help to make up a lot of things in the bone marrow, they're osteoblasts. These are the young version of what is to become osteocyte. The bone cells, osteocyte, they help to mature the bone. If you wonder why you have minerals and all of these things, the osteocyte help to produce those. Osteoblasts are the immature version. They secrete the compound or the matrix or the extracellular matrix. That's important to know because you will get to the level of connective tissue. Understanding connective tissue, understand that there are different types of connective tissue. For example, here, the osteoblast is responsible for producing a lot of the substance that are used to make up the connective tissue in the bone. And of course, you also have progenitor cells, which are, as we said, the blast cell. And you also have some multinucleated cells that help to secrete enzymes and acid that help to dissolve the bone itself. So even in medicine, you'll find that there are some pathologies where there is breakdown of the bone. It's the osteoclasts that are there just breaking down stuff. So in reality, I always say there is nothing without an answer. There's always an answer to everything. And as a molecular person, you really have to believe that, because sometimes when you really go to the root of the problem, the genetic information can tell you a whole lot. All right, so we can look at the skin cells now. The skin cells, we know layer of the skin is epidermis. The cells are the keratinocyte. We also have different types of cells like white cells in the skin, nerve cells. You have white blood cells in the skin that helps to fight infection. You need it everywhere. You also have nerve endings or nerve cells in the skin. So the skin is just a combination of cells. Everything in your body are a combination of cells. It's a matter of how you are able to dissect or look at them individually or look at them by detail. So even with this, for the layers of the epidermis, you have different layers like the stratum corneum, lucidium, granulosum, spinosum, and each layer you have different types of cells and the distribution of certain cells. For example, you know that cells die on your skin every day, right? Anybody can give me like the random answer to this? How many cells you regenerate per day, or how many cells, dead cells, fall off of your skin every day on average? So Google answer.
30,000 to 40,000 dead skin cells fall off your skin, your surface of your skin.
Okay, I thought it was more, but I won't fight with Google.
Rihanna,
okay. Oh, thank you for that knowledge, Rihanna. Why you have so many cells or keratinocytes falling off your skin each day? Naturally, they're made at the base, the stratum basale. You have the nerve endings. So if I touch you, you're only able to feel it because you have the nerve cells in at the base layer of the epidermis itself. You also make these cells that are supposed to line your body, keratinocyte, and when they're created, they slowly rise all the way to the top. So if you look at from the bottom to the top, you realize that the living cells are actually at the bottom, and at the top, pure dead cells. So if you're even in the palm of your hand and the sole of your feet, pure dead keratinocytes, and they're falling off each time. So as I said, the keratinocytes are produced by stem cells in the stratum basale, the base, the lowest part of the epidermis. And once a keratinocyte moves more than 3 cells away from the dermis, it moves up and the mitosis stops at a certain point. But you're always creating new cells from the bottom. All right, and then we go into the muscle cell. So for the muscle cells, you have different types of muscle cells. You have different types of skin cells. Now you have different types of muscle cells. So you definitely will need to know the difference between the different types of muscle. For example, skeletal muscle versus smooth muscle versus cardiac muscle. Could anybody give me an example of a smooth muscle in your body? Yes, there's smooth muscle in the intestines. Anywhere else?
the bladder.
Yes. And we were all created in a smooth muscle. The uterus.
The esophagus.
Yeah, there's also smooth muscle there. All right. So the skeletal muscle, as obvious, it lines like the bone. So basically, all the muscles in your hands, and you have a lot of muscle, like even in the forearm. They all have different names. Like, I couldn't even want to list them right now. But all of these muscles that are attached to tendons that are attached to bones, they're skeletal muscle, and they're different histologically from skeletal muscles versus cardiac muscle. And where do we find cardiac muscle? In the heart, exactly. So skeletal muscle, and this is the histological feature. You can know this right now, and this will definitely be like a question in your final exam, because the whole point of learning cells, learning tissues, learning muscle is to learn the difference between things. That's the whole point. If they all were the same, we wouldn't need to be learning anything right now. We'd just say every cell have some fluid in it, and it just behave the same. For skeletal muscle fibers, they're striated. You understand that eventually. Know that they're tubular and they have more than one nuclei. They're multinucleated, and these are voluntary muscle cells. They're voluntary. You decide when to move, you decide when to punch, you decide when to walk, you decide when to run. These are voluntary muscles, and they're always attached to the skeleton. Smooth muscle are non-striated. So know the difference. What is striated from what is not striated? What is the shape? One is tubular, smooth muscle is spindle. It's uninucleated. And the difference between that and skeletal muscle, multinucleated. So the smooth muscle tend to cover the internal organs. You find them in the wall of internal organs. They're covering most of your organs. And who could tell me where the smooth, what is the embryologic origin of a smooth muscle, for example? Endoderm, mesoderm, or ectoderm?
Will be endoderm. Endoderm.
And did Google give you the answer?
I was saying endoderm.
Are more like mesoderm. All right. So the cardiac muscle fibers are striated. That's the difference. So skeletal muscles, cardiac muscles, striated. They're brand and uninucleated. So only skeletal muscle have more than one nuclei. The cardiac muscle is involuntary. Obviously, if the cardiac muscle was voluntary, a lot of people would be alive right now, right? If you could just voluntarily make your heart beat, you wouldn't let it stop, would you?
No.
And the important thing to remember with this cardiac muscle is that it has what we call intercalated disc. Intercalated disc, you will see this on histology. So if I show you anything about muscles, I'll show you probably 3 different histology slides, one with skeletal muscle, one with smooth. And if you look to the right, the extreme right, this is the arrangement of the muscle fibers. And you can see also the nuclei in one. You can see 1, 2, 3 going all the way down. And if you look, for example, the cardiac muscle, you'll eventually see that they have what is called intercalated disc. And you will need to know that cardiac muscles have intercalated disc. What is the function of the intercalated disc? In terms of the skeletal muscle fibers, just know that the muscle fibers in general, they're arranged in groups, sometimes in fascicle, and they're wrapped by fascia, like a thin lining just wrap around a group of these cells that create fascicles. Of course, all of those are wrapped around perimysium and the epimysium. I won't ask you too many complicated stuff about this as it relates to tissues and compartments. Cardiac muscles have intercalated disc. And if you look at the extreme left, you can see the nucleus here. Is it multinucleated? Was it?
It was uninucleated.
Okay, and you have the intercalated disc, and this is very specific. So if I show you a slide or just a photograph and it's a muscle, you don't see anything at all, but you see these things that are branching across the cell itself. They're interrupting. They're called intercalated discs, and you should know that intercalated discs are only specific for the cardiac muscle. All right, so these are fat cells. You can have brown adipocyte, white adipocyte. Brown fat and white fat is the most important to remember. Brown fat versus white fat. Why? Brown adipocytes are found in mostly animals, and you know that the adipose tissue is there for insulation, make you feel warm. If you look at a histology, for example, of an adipocyte, you will see that it's pale. It's usually pale because of the fat in it. The nuclei just push to the end, or the rim, or the edge of the cell, or the nucleus is just pushed to the edge of the cell. So this is just showing brown fat and energy conversion. So this is a histology. This is a slide of a fat cell, clear cytoplasm. And if you look at the edge, you can see the nucleus squish to the edge. So if I show you like a slide of this, should be easy. And for the neuronal cells, you have different types of neuronal cells in the central nervous system as well as the peripheral nervous system. The glial cells or neurological cells are support for the neurons. So they support and protect the neurons. And the neurons themselves are the nerve cells that receive the stimuli. They transmit action potential. They're organized. They have a cell body, just like you have the big cytoplasm with all the organelles. You have like dendrites that receive the action potential, and then you have the axon. It sends the signal to certain place. Example of cells, neuronal cells, you have astrocytes. These cells, all cells can cause their own disease. So it's not only the organ that can be diseased, it's the cell. And if you really look at the genetic structure of the cell and you can extract some genetic information, chances are there's some genetic mutation along that cell line, which is why people tend to have certain problem of the cell. For example, astrocytes are cells found in the central nervous system. Sometimes you have what we call astrocytoma. So like it's a tumor that you see in children, and it's basically as a result of the astrocyte. So the microglial cells, you have microglial cells as well as oligodendrocytes. All of these cells have different function. You will get to understand eventually, but this is just basically a synopsis of what they are and their function. And certain disease, for example, in Alzheimer's disease, this is just basically giving an example or making a medical correlation as to what happens to each of these neuronal cell components in this type of disease. So I would say know this. Like just have a basic understanding or idea as to, you know, the neuronal cell itself, the nucleus, the cell body, the dendrite, the axon, their function. So you have different types of neurons, can be unipolar, monopolar, bipolar, pseudo-unipolar. We should cover that in a little bit more detail eventually. Yeah, and we already went through sex cells. The basic structure of the cell almost always the same. There's a mitochondria, there's a nucleus, there is the cell membrane or plasma membrane. This one has a flagellum. The human egg, the zona pellucida is that coat that is around the cell. Then you also have a zone around that zona pellucida that is also protective. Sperm will have to pass through all of that to get to the nucleus in the cytoplasm. We already touched a bit on endothelial cells. Pancreatic cell is just to add a little bit of medical correlation to different types of cells in the body. For example, in the pancreas, if you know the different types of cells in the pancreas and how they're affected, you know what's happening to the patient. For example, the beta cell secretes gastrin and increases the insulin production. So, for example, patients with, if somebody say they have type 1 diabetes, it's because the beta cells are damaged in the pancreas. Because you can see the beta cells are here to increase the insulin. These patients are insulin dependent simply because the beta cells are damaged, not producing the insulin. There are a lot of other diseases that are associated with destruction of cells in the pancreas, but the pancreatic cells are unique because you don't find them anywhere else in the body. And the pancreas is a very important organ, as much as it's that small and seem irrelevant. Notice that when people have cancer of the pancreas, they tend to just die quickly. Anybody remember her name?
Doreen Summers.
Yeah. She basically died quickly of pancreatic cancer. And this is probably not the first time or the second time you'll hear this. You'll probably hear this sometime again in your lifetime. All right. So, and then we go to the tissues. Of course, the cells make up the tissues. So you have the connective tissues that are wrapped between, for example, the epidermis, the epidermal cells, the keratinocytes. For each layer, there are connective tissue intermixed. So the connective tissues are there, of course, as support. There are protein, there are the cells that create the surrounding connective tissue structure. The chicken is soft because you break down the connective tissue. So when you cook the chicken, when you boil the chicken, when you throw it in the pot, you break down the protein, you're damaging or causing denaturation of the protein, and that basically separates the connective tissue from the muscle, from the other structural components, so that you can bite the chicken. You could bite it if it wasn't cooked. So the connective tissue is a very important component of everything. The muscle cells obviously together create the muscle tissues. Nervous cells together create the connective tissues. Epithelial cells create the epithelial tissues. Tissues or connective tissues can have different arrangement depending on arrangement of the fibers and the substance that are in it. Connective tissue can be dense, it can be loose. You can have loose connective tissue, you can have dense connective tissue. And as you can see, there's connective tissue in everything. The adipose tissue, areolar tissue, in compact bone, there's also connective tissue. There's connective tissue in the blood. So as I said, the connective tissue can vary based on the arrangement of the collagen fiber. So eventually you'll know the composition or the constituent of connective tissue, whether it's a loose connective tissue. What is the difference between a loose connective tissue and irregular connective tissue? Loose connective tissue, the collagen fibers, they arrange. So yeah, the loose connective tissues, the collagen are arranged, the fibers are arranged in a different way. You also have elastic fibers and fibroblasts. So for each connective tissue, you'll realize that there is different types of collagen in each connective tissue, and there are different cells But as you can see, there's always fibroblast, connective tissues in general. And this is just like a histology of loose connective tissue looks like. I don't want to put this on a slide and you tell me it's a cardiac, it's a muscle cell, right? With intercalated discs. So we already touched a bit on the embryology, which is the end of the presentation. We already touched a bit on the embryology. We know that after the fertilization, you have the zygote, the 2 cell to the 4 cell to the 8 cell to the blastocyst, and each cell differentiate to create endoderm, mesoderm, ectoderm, and each of these endoderm, mesoderm, ectoderm is responsible for the differentiation. I think that's the end of the presentation. Any questions?
No questions from me.
Okay, great. Okay, so that is it. I will make these things available online so that everybody can have access. Feel free to email me if you have any questions, any issues. Session today was basically everything that you will know a little bit more about. So it's nothing to be alarmed about. It's not going to be complicated.
So do you have like a course outline that we could use?
Yes, so the same. You guys don't have the same course outline, right? You guys have a course outline from... No, you wouldn't. All right, so I'll just upload that course outline. I didn't really make mention of much of it because the content is basically we're guided by the content, which is everything that was on this slide. On that course outline, there was no provision for like lab sessions, which is why I didn't really pull it up. So I'll try to tailor this course outline to be more specific to what you guys are required to know, and I'll upload it as well with a PowerPoint.
Okay, thank you.
All right, so see you guys next Wednesday. So I'll make that link available sometime this week or next. And if you have any questions in the meantime, if you need guidance, you can just email me at the UMONA. I should be able to respond hopefully in less than 2 days.
Just could you send your email in the chat?
Sure.
Okay, thank you.
No problem. See you guys next week, Wednesday.
Okay, bye-bye.
Bye.